Hello. Hello. Whoever wants to ask a question first, I'm ready, willing, and hopefully able.
That you've heard about it?
Hi.
Hi.
I was wondering if you can kind of talk about the oncohistones again. Just talking about how they, explaining how they like poison epigenetic regulators.
Yeah, sure thing. Let me pull up those slides and I will share my desktop. There we go. Play and no, I do not want that. Sorry, I have to set this up a little differently. Here's the keynote play. Alrighty, I should do better. And then all I gotta do is share my screen with you. There we go. Okay, can you see my screen?
Yes.
All right, let me blow that up then. All right. So, oh wait, there's a couple of people who probably need to be let in. Hold on. All right. I can't see who's in and who's out, but okay. So we have we started with a couple of examples of different changes that occur in different kinds of cancers at various histones. But I think the part that you were most interested in was this part here. And let's just focus on this example because I think the other example just confuses things. And this is the one that I want you to try to remember, keeping in mind that there are probably other mechanisms for changes, mutations in histones that could be involved in cancer. So what happens here is that you have a change. So here's histone H3. Actually, let's start here. You have histone H3. Here's the tail. And normally, the enzyme that's the histone methyltransferase, which is called enhancer of zest in humans, there's 2 copies of it. This will add 3 methyl groups to lysine 27 on the tail of histone H3, right? So under normal circumstances, that will convert this from no methylation or monomethylation or dimethylation into trimethylation. Now, what happens in the mutation is that when you mutate that lysine to a methionine, then what happens, there we go, mute all again. Then what happens is that, so now this is supposed to indicate the filled oval indicates that you have this mutant histone. And remember, these are acting dominantly. You have normal histone H3 in the background, which has the potential at least to undergo the same kind of trimethylation. So what's happening here is that by having this methyl group here instead of a lysine, for whatever reason, I mean, it's a structural issue, the catalytic site that binds to that, when it binds that peptide that has K27 in it, it binds it with much higher affinity than it normally would, okay? And that so the way that this works is that it essentially sequesters or holds so tightly onto this enzyme that it can't leave and then go to these other sites, even on the normal histone H3s that have a normal K27 instead of a thymine. It can't leave or doesn't leave as efficiently, so it does not methylate those residues. And so what happens is you get a global decrease in the levels of this mark, which is associated with polycomb binding, but most importantly is associated with gene transcription suppression. So normally you would suppress all these genes using polycomb and enhancer of zest and all those things, but genes that are, sorry, along the DNA here. But in this case, what you're doing is you're holding on so tightly to the enzyme that it can't leave or can't leave as efficiently. And so you're not getting normal silencing. And so think about if there's an oncogene somewhere here, problems arise with oncogenes when you overexpress them. And so that basically means that you're probably getting overexpression of one or more oncogenes and that's contributing to cancer. Does that answer your question, Jennifer?
Yeah, thank you.
Okay. It's complicated. The other example I'm not even going to go into because it's it's even worse. So yeah.
So which model does this
I can go.
follow?
What's that? Sorry.
So I
Which model does it follow the genome instability?
Codify a lot of
Sorry,
the
I'm having.
UAC does by writing up.
Sorry, Abby's talking in the background. Sorry, say that again.
I was just wondering, like, which model it follows. Is it like the genome instability or
this exact slide.
the?
Sorry, I'm hearing someone else now too. Which model?
Genome instability versus oncogenes, since there's mutations in the oncohistones.
Oh, well, you can't really say for sure. I mean, you could also end up altering the expression of genes involved in DNA repair, chromosome segregation. It's not known at this point. It's not known, at least I've not seen published work related to related to why these cancers are so aggressive and especially impact young children. So yeah, it's not necessarily, this could lead to genome instability. It could lead to oncogene expression, overexpression, or misexpression of tumor suppressors. There's all sorts of possibilities here that haven't been looked at. We're focusing on transcription because that's a known function of polycomb.
Okay, thank you.
I am, of course, happy to answer any questions about any of my lectures. I can even try answering questions about the other two lectures, but you're better off asking them. No one? You can also type your message in the chat window. I finally figured out how to be able to see all of those at the same time.
I had another question.
Mm-hmm.
So what exactly does it mean for the genotype to become very heterogeneous? Is it just that the multiple like genotypes, or like there's multiple, there's a lot of different kind of cells or mutated cells in like a tumor?
Yeah, so this goes back to, well, we showed this in a couple of different ways. Here's one version of that. There's an earlier slide showing this, and it came at the very beginning and might make more sense now. But what we're talking about here is heterogeneity among cells in their genotypes within a tumor. So you have many cells in the tumor, and you have many different types of cells. But even among the like epithelial cells versus other types of cells, but even among just the epithelial cells, or those cells derived from epithelial cells, because there's a lot of mutation going on compared to a normal cell lineage, you know, each of these steps, probably there's different mutations or rearrangements or things like that. In the end, you get what are really different genomes in this cell versus these cells versus these cells. And so that's what we mean by heterogeneous, that there's homogeneous would be that all the cells are the same, so they're all orange in terms of their genomes. Heterogeneous means that there's a mixture. And so the reason that this is really important is that selection of any kind can act on these cells, and you don't need all the cells to respond in what for cancer would be positive ways, meaning unrestricted growth, metastasis, invasiveness, etc. You can only select, you're able to actually continue to progress or become even more extreme of a cancer by having this variation and then letting selective agents selection act on those cells, so that, sorry, this one's not a very good one, but well, you can look at this way. You have, you know, this is would be what a tumor would be with all these different cell types. You still have some of these cells, even among these cells, you would have mixtures of different intensities of blue, let's say, and they all have different properties. This cell here is actually invasive, this cell metastatic, this one doesn't need as many growth factors anymore, et cetera, et cetera. But once you apply, let's say, a drug Maybe even if 99.9% of their cells, of these cells get killed, if there's one whose genome has been mutated in a way that makes it resistant to that drug, then it will continue to divide and recreate a tumor or metastasize, that sort of thing. So this is a huge problem in treatment because as a result of this heterogeneity, you can kill off most of a tumor with some kind of chemical agent, and yet it will come back months later or years later, simply because you didn't kill all of them. And so this is why people use combinations of therapies and epigenetic therapy plus Taxol or cisplatin, those kinds of things. Does that help?
Yeah, that helps. Thank you.
And then there were 3.
Comment.
You guys have to have some questions. You weren't all going to just lurk, were you? Yeah, I know. Thanks, Al. I appreciate that. And yeah, you don't have to ask questions. But unfortunately, when only a few people show up, lurking doesn't work either. If there aren't that many questions. Maybe Julia has a question. Not to put her on the spot, though. I could just show you our new puppy if you prefer that.
Hi, Professor. Did you talk about what GIN and CIN is?
Yeah, so I didn't emphasize that, and I find them to be annoying terms, but generally speaking, and I prefer it this way. So can you see the slide here?
Yes.
Yeah, okay. Where's this going? Okay. Yeah, so when people talk about GIN and SYN, so I think the easiest way to think about it is that genome instability is a general category that includes things like small changes, mutations, single base changes, maybe a small insertions or deletions. So what we would normally call mutation. Chromosome instability is much more specific, meaning it has to do with changes in the number or the organization of chromosomes. And so all of those rearrangements that we talked about when we showed pictures like this, those would be considered SYN because they involve chromosome changes. So the scale is much larger than the sort of small scale of mutations or small insertions or deletions. So chromothripsis would be an example of that. The telomere shortening and the major changes in chromosome number, chromosome type that result either from telomere fusions and then dicentric formation and then things being pulled apart, and or just changes in ploidy. Like sometimes you get in cancer cells, they undergo replication, but they don't actually undergo cell division. And so they go from 2 copies of every chromosome to 4 copies of every chromosome. And then there are all sorts of changes in the amounts there. So anything at the chromosome level in terms of changes in the number or the organization of the chromosomes would be considered SIN. Genome instability is a more general term, which includes mutations.
Hi, Dr. Karpen.
Mm-hmm.
I was wondering if the material on the exam, at least for your portion, will it be mostly concerned with the cancer stuff that you talked about, or like the stuff that you taught in your first section?
It'll be both.
Okay.
I would say that I will, because I haven't asked questions about the cancer stuff, I will, I don't know, I don't want to say emphasize more, but I think there will be more content related to that. It might hark back to previous lectures, for example, talking about all the epigenetic kinds of changes. You know, I might focus it on cancer, but then hark back to a previous concept that we discussed. Yeah, I mean, I'm limited to a fairly small number of questions, so
Yeah.
but I do, you know, I think first of all, I think it's fair to focus a bit more on what we just talked about. Also because we haven't talked about it, but you know, we talked, for example, if you look, you know, we talked about writers, erasers, and readers, right? So,
Yeah.
you know, I'm not going into detail about those here. I didn't go into that much detail before, but at least, you know, what our discussion about, I don't know if you were on earlier, we were talking about the oncohistones, you know, the whole idea about
Transferase stuff, right?
yeah, that, I mean, this sort of only makes sense if you're aware of that this is a writer, there's a reader in here somewhere, and you know that if this methyltransferase can't leave as readily as it normally does, then you will lead to less and less trimethylation at other sites. And so, you know, you can understand that a little better, I think, in the context of what we discussed back a couple of months ago, way back in January and February. But, you know, this is also what we didn't discuss then in any, I don't think I mentioned it even at all, is that because these are enzymes, so enhancer zest and the suvar39, as you know, the 2 ones that I have emphasized, ignore SETDB2 or SETD2 or SET1, but you know, these 2 methyltransferases, they're not present in very high copy number relative to the readers, and of course relative to the histone targets themselves. So, you know, this wouldn't make sense as a mechanism if you had so much of this enzyme that it didn't matter that, you know, 1/10 of it was stuck here. 90% of it could go elsewhere. And, you know, that does happen because this is dominant. It could be that only 1/10 of the histone tails have this particular mutation. but it's enough to cause a decreased silencing of other genes.
Mm. Thank you, Professor Karpen.
Sure. I think as Nicole said, since we're on the topic of the exam, I mean, it's mostly driven by Abby, but I'm sorry, Dr. Dernbit, but we recognize that things have been quite difficult for everybody, and we're not looking to make things harder. Let's put it that way.
Thank you very much.
But yeah. And if you would ask me, what do I consider to be most important within the cancer lectures? Would you like me to tell you?
Yes,
The
please.
You're going to be rewarded just for showing up. No, so okay, oncogenes and genome instability. I presented that as these alternative mechanisms. I want you to understand the difference between altering growth control at the sort of cellular level and all this sort of cellular things that have to happen to be metastatic and things like that, and what's happening at the genome level. And the bottom line is, there's both, and you can't be a bona fide cancer cell if you don't have both genome instability and changes in oncogene expression or mutations in oncogenes or tumor suppressors. And the bottom line is that most late stage cancers have all of these things and then some, right? There's one slide on DNA repair defects. I'm not going to emphasize that. I emphasize the sort of chromosomal abnormalities and the genome instability because basically for the last 30 years, most people have only talked about oncogenes and tumor suppressors. So it was always from the, it's been emphasized that changes at the individual gene level are what matters. And, people are now coming back to what was known 100 years ago, which is that there's a lot of genome instability and that plays an important role. For telomeres, I just wanted you to understand that they have to do really two things for cancer to be generated or progress, and that is, they have to shorten. That's a natural part of somatic differentiation, right? It's just as cells age, they stop making telomerase, the somatic cells, not the germ cells. So you shorten them, and that shortened telomeres lead to instability because they start to be recognized as double-strand breaks, and normally they're not. So once you're recognized as a double-strand break, then DNA repair comes in, and normally we think DNA repair is a good thing, but if you fuse two telomeres together, then you're also fusing two centromeres along that same molecule, and that means that you are now creating genomic abnormalities that will generate even more genome instability. So, the role of telomeres here is, number one, generating genome instability, but then they have to go through this second round of regulation, which is that, okay, you've generated all this genome instability. If that keeps going, you're just going to kill that cell. It's going to become so aneuploid that it's not going to be able to be a cancer cell. Cancer cells still have to function, albeit abnormally. So you have to add telomeres back to stabilize that genome in whatever form it's in at that point. Of course, it's only stabilizing it in terms of telomere fusions. You're still getting mutations, and that's where you're getting all this heterogeneity arising as well. But it's this weird sort of, I don't know if it's yin and yang, but at least you've got to shorten them in order to generate the genome instability, and then you've got to put them back on either by reactivating telomerase or doing this alt recombination mechanism. So, why is that? Because normally you would get these short ones giving rise to apoptosis and aging related phenotypes, not cancer. So you have to make them long again in order to overcome that part. So, you know, telomeres, again, you can see I'm talking about sort of the basics of where these things stand, you know, questions, which are great questions about, you know, the alt mechanism or how does telomerase get reactivated. You know, we can talk about that, but I'm not asking you to understand that. And sometimes we don't really understand it. Yeah, so chromothripsis, I think I spent some time on, you know, I think there, again, I want you to understand the logic that you have a genome, a chromosome segregation defect that makes some chromosome lag behind or a piece of a chromosome lag behind, and it stays at the metaphase plate. And that leads to formation of these micronuclei. So as cells come out of mitosis, the nuclear envelope reforms. And normally it would reform around all of the chromosomes as one nucleus, but if you're left behind, you're left behind and you're not incorporated into the major nucleus. And then, and, you know, the bottom line is that even though chromothripsis is not necessarily the most important mechanism in genome instability in cancer, I'd say telomeres are in terms of their general impact across probably all cancers. But that, you know, this is a very interesting way that the cell biology intersects with genome stability, and in a way that gives rise to multiple changes, not across the genome, but in a single chromosome. And then, you know, in terms of this sort of blessing or curse thing, I wanted you to understand that
Your cells need some instability, but they can't have too much, right?
yeah, exactly. So this sort of, you know, that's a theme that was emphasized in other ways, like the whole, if you don't cap your telomeres again, then you're going to get up here, and you're going to die. And you know, oddly enough, sometimes cell death is a good thing for organisms. But yeah, if you're down here, even with oncogene mutations, so you might continue to grow, but it's unlikely that you'll gain the mutations or the genome changes that will give rise to metastasis, the more advanced stages of cancer. But yeah, so you want to sort of live in this zone where you have moderate genome instability. And so that,
Cancers,
you know,
though.
what's that?
For cancers, right?
Yeah. Sorry. Yes. Thank you for that clarification. Yes. Yeah, as you see at the top, too much instability is bad for cancer cells. It's bad for normal cells too.
Yeah.
Which is why, you know, we talked a little bit about therapeutic, what I would call sort of broad therapeutics, meaning they don't target a specific type of mutation, like a specific kinase involved in melanoma or things like that, but they just broadly either cause DNA damage or cause chromosome segregation defects. Those things are just going to kill, are going to cause mutations or chromosome rearrangements or chromosome loss in all cells that are dividing. And so that's why you get side effects with hair falling out and gut cells. So any and blood cells as well, actually. So anemia is common, and there are always there are good ways to counteract that, but they're never perfect, and it varies from individual to individual. So yeah, I mean, these are acting by pushing the cancer cell to here, but they're also doing it to other cells, and that's problematic. And then the last thing is just, you know, epigenetic alteration. I mean, the oncohistones are an interesting case, as we talked about earlier in office hours. Mostly, I wanted to just point out that, you know, if you think about mutations, yes, there's a lot of mutations that will change growth control genes. But a lot of these epigenetic regulators, including polycomb enhancer zest, are regulating normal growth control genes or repair genes or things like telomerase genes. And so that, to keep in mind that epigenetic alterations also can impact cancer at many different levels. And I think I used this as an example. I didn't go into detail here, but just, if you go back and look, a lot of these are mentioned in the sort of broad hallmarks of cancer. And these are just examples where an epigenetic change is involved in specific cancers in causing abnormalities in these kinds of stages or barriers to cancer. So that's, basically with broad strokes, what I think is important in the cancer lectures.
Thank you very much.
I didn't go as far as giving you the questions.
Yeah.
That might be too unfair, but it's all right. All of this is being recorded and will be put up anyway, so everyone will have a chance to see it. Whether it's going to help anyone or not is another story. All right. Any more questions?
Could you talk about what polycombs are?
Polycomb?
Yeah.
How far back do you want me to go? So are you asking just why is it called polycomb, or are you asking about sort of the whole mechanism?
Like what it is and what it does.
Okay. I'm trying to think if I should go back to. All right. So, polycomb itself, it's named that because it was discovered in flies, as a lot of these epigenetic regulators were, and it changed body plans so that legs that normally didn't have what are called sex combs all of a sudden got sex combs. So that's why it was called polycomb. You won't be asked that. But it's unfortunate that name doesn't really help you when talking about cancer and all that sort of stuff. What its normal job is to set body plan. So we, as is true for flies, are segmented organisms. We have a head, we have a thorax, we have all these vertebrae, right? And so in flies, as well as humans, there are these genes called homeotic genes that become activated in certain parts of the body, and they will, for example, change this segment to have arms, and the lower segments not to have arms, right? And they do this through gene repression. So essentially, as you go from the anterior of an organism to the posterior, you have a lot of genes expressed here, and then as you go posterior, more and more genes are repressed of these homeotic genes, which are just transcriptional regulators, either repressors or enhancers that act on developmental genes that will make an arm or a leg or anything like that. Okay, so the bottom line is that the polycomb complex is responsible for adding this H3K27 methyl 3 mark. So think back to the epigenetics lectures where I emphasized H3K9 methyl 2 methyl 3. That involved transcriptional repression as well, but they're distinct in terms of the components and, of course, which part of the tail is modified. So you have the system for what is called facultative silencing, or unlike the constitutive heterochromatin, the K9 methylated chromatin, that's always there. It's always over these repeated sequences. This developmentally regulated and will occur over some genes and not others, and obviously in a regulated pattern. So this polycomb complex, it's all about adding this mark, and then this mark will be bound by the polycomb protein, which is like the HP1 protein, heterochromatin protein 1. It has a domain that lets it specifically bind this H3K27 when it's trimethylated. Now, it turns out that in addition to its role as a developmental regulator through silencing genes that, for example, are responsible for making an arm, you want to silence them lower down in your chest, in segments made from those parts of your body, because you don't want 6 sets of arms. Or maybe that would be useful at some times, but most humans really want to just have 2. So polycomb is what binds it, and enhancer of zest is the enzyme that adds that methyl group. Now in cancers, what you have is a lot of the polycomb and enhancer of zest, all the things in this complex are considered to be tumor suppressors. And that's because they are involved not only in repressing expression of body pattern genes, but also things like growth control genes. Those are part of developmental regulation as well. To make an arm, you need some cells to divide and other cells not to divide, things like that. So it turns out that polycomb, enhancer of zest, all these components are also regulators of oncogenes and also, in some cases, other tumor suppressors. So its importance is that it's another epigenetic system for silencing beyond the K9 methylation HP1, and it seems to play a particularly important role in cancer. It's not just about these oncogenes, it's also, there are other cases we didn't talk about where individual components of the polycomb complex themselves can be mutated, not the histone amino acids, and that those can also be observed as mutated in different kinds of cancers. So keep in mind, this is a very specific model for why these oncohistones have impact. But behind that, there's they're related, they're not unrelated. In this model, it's the loss of silencing of other genes elsewhere in the genome, specifically oncogenes that are silenced normally by this H3K27 methyl 3. But it's the loss of that silencing that contributes to cancer. Does that?
Yes, that helps. Thank you.
It may have been more than you wanted, but or needed, but at least, yeah. Anything else? Should we call it a day? Let's see if I can find the puppy. No, puppy's gone.
Thank you very much, Professor Karpen.
Sure thing. Good luck. I'll be back on Wednesday if there are any other questions.
Yeah, I have your office hours written down, so I'll probably be there for your next one.
Okay, good.
All right.
Good. Take care.
Thank you. You too.
Okay, bye.
Bye.